I should have already, and I'm not sure why I didn't. I've been wanting to meet you all, so this is very exciting for me. And I will talk about local research. I'm going to put research in quotations. This was really born out of, it is research, of course, I should have
it's
already,
very
preliminary.
Sure I didn't.
So
I've been wanting
what
to
I'll talk about
meet
today
you all,
was born out of
so
a
this is
project that I did with my I will talk about local research. I'm going to put research in quotations.
And then what
really
happened was I had 7 students that were so excited about what we were doing in class.
So what I'll talk about today
volunteered
was born
to
out
work
of
in
my
my
project
lab.
and so during the summer we collected additional
pollination
data.
biology
And
class.
I've been using this sort of as an exploratory
And then what happened was I had 7 students that were so excited about
wild
what
plants
we were doing in
pollinators
class.
around
They
here.
volunteered
And
to
so
work
we've
in my
kind of
lab.
documented
And so during the summer, we
a lot of
collected
this
additional data.
correlational.
And
I've been
really
using this sort of as an exploratory process to get to know what kinds of things you think might be driving the patterns.
Pollinators around here
too. And so we've kind of documented
anything that
some
excites you that might be
a lot
good
of this
for
is going to be
future
correlational
research with
and I'm
classes
really
or,
excited to
in the
hear
lab.
from
I
you.
like to
In terms
have
of
a local
what kinds
system
of things you think might be driving the
we're so
patterns.
fortunate
I
to
have
have
some ideas too.
such a robust
and then anything that
ecological,
excites you that might be good
system
for
here in
future
the Bay.
research with classes
So,
or
in the lab.
I will start by
have
kind of
a local
conceptualizing
system
the
and
work in the way that
we're so fortunate to have
do in
such
my
a
lab.
robust
As you are all aware,
system
we live
here
in this era
in
in
the
which
Bay.
human-induced environmental changes are imposing.
I will start by
kind of conceptualizing the work in the way that from the breaking up of habitat into small remnant fragments. As you are all aware, we live in this era in which human-induced environmental changes are imposing
familiar with that
a whole suite of challenges on organisms. From the breaking up of habitat
non-native
into small
species and
fragments,
genotypes to areas that were once
to
devoid
climatic
of them,
changes that are inducing
drought. We have
really
extreme
drought
pollution.
pollution.
Everyone is familiar with that in
And
California.
I'm an evolutionary ecologist, so
to,
I'm really interested in
the
how
introduction
these
of
environmental
non-native
changes
species and
shape
genotypes
biological
to areas that were
diversity.
once devoid of
So
them.
that's sort of the big question that I
and
asked
to
in my lab.
really
And
extreme
then
pollution.
I really
in some
started
areas.
thinking recently
And
about
I'm an
this
evolutionary
other question,
ecologist,
so I'm really
which is
interested in how can
these
we increase
environmental
positive
changes
changes
between
shape
human-dominated
biological
and
diversity.
natural areas.
So that's sort of the big question
And so
that I asked in
to
my lab.
get at both of these
And
questions,
then I really started thinking
study
recently
interactions
about
between
this
plants
other
and
question,
pollinators.
which is how can
And of course,
we
we're
increase
all
positive
pollinator
feedback
enthusiasts
between
human-dominated
here,
and
but I'll still
natural
explain why I
areas.
use
interactions
so
between
to
plants
get
and
at both
pollinators.
of these
To
questions,
get at these
I
questions,
study interactions
and
between plants
I
and
think that
pollinators.
plant-pollinator interactions are one of the best
And of course,
ways
we're
for
all
studying these
pollinator
questions
enthusiasts
because
here.
the
But I'll still
interactions
explain
represent
why I
100
use
million years of
interactions
evolution,
between
yet
plants and pollinators
the
to get at these
interactions really lack
questions.
specialization
And
in most cases.
I think that
which
plant-pollinator
makes the
interactions are one
interactions
of the best ways for
context-dependent.
studying these questions.
And that allows
because
us to study how
the
subtle
interactions
changes
represent
100 million
in
years
ecological
of
conditions
evolution,
might
yet
actually change the outcome
interactions
of
really lack
interactions.
specialization in
And
most
the
cases.
other reason I
which
think
makes the
plant-pollinator
interactions
are so good for getting at
context-dependent.
these questions,
And
and
that
I wonder if you would
allows us to
agree,
study how subtle
is
changes
that
in
I really think
ecological
strengthening
conditions might
pollination
actually
systems is one
change
of
the
the best
outcome
ways
of
to increase
interactions.
positive feedbacks between
And
human-dominated
the other
areas
reason
and
I
more
think
natural
plant-pollinator
areas.
interactions are so good for getting at these
So
questions.
what do I mean
And I
by
wonder
that?
if you would agree.
Well, of course,
Is
pollinators
that
facilitate
I really think
our being able to
strengthening
eat healthy
pollination
food,
systems is one of the best ways.
and we really
to
need a
increase positive
diversity of
feedbacks
pollinators
between
in order to
human-dominated
have
areas
healthy
and
food
more
that we
natural
want.
areas.
So, of course, the tomato
So
flower
what
above
do I
needs a
mean
buzz
by that?
pollinator like a bumblebee.
Well, of course,
and then
pollinators
that peach flower
facilitate
below
being
can be
able to eat
pollinated
healthy
by
food.
a honeybee.
And we really need a diversity of pollinators.
And then, of course,
in order
pollinators
to have
facilitate the
healthy food that
persistence
we
of
want.
wild
So,
plants
of course,
ranging from cacti.
the tomato
and,
flower
this
above
is
needs a buzz
broader than
pollinator
just
like a
bees.
bumblebee.
So that's
and then that peach
a
flower
bat
below
pollinating
can
that
be
saguaro,
pollinated by a honeybee.
ranging all the way from cacti to these delicate wildflowers.
And then, of course,
That's
pollinators
phlox on the
facilitate the
bottom
persistence
right.
of
And I actually
wild
study
plants
all of
ranging
these
from cacti,
systems,
but
but
I'm
this
going
is
to talk with you
broader
about,
than just
mostly,
bees. So that's
bees and
a bat
flies in the Bay
pollinating
Area
that
today.
saguaro, ranging all the way from cacti to
And
these
since we know
delicate
that wild
wildflowers,
bees
that's
will
phlox
pollinate
on the
agricultural
bottom
crops
right.
and
I
actually
study
even
more
more
effectively than
honeybees,
honeybees. So
I'm going to talk with you
study in
about,
Journal of Applied
mostly,
Ecology found that
wild
bees
bees
were
and
more
flies in the
important
Bay
than
Area
honeybees.
today.
It's really important, of course,
And
to
since we know
understand
that
how these
wild
environmental
bees will
changes affect
pollinate
wild
agricultural
pollinator
crops and possibly
populations,
even
and
more
what types of
effectively than
interventions might
honeybees. So
augment
this study
abundance
in
of
Journal of
wild
Applied
pollinators.
Ecology found that
And of course,
wild bees were more
interventions would
important than
also
honeybees
benefit from managed
it's really important, of
honeybees.
course, to
And
understand
I'm
how
a
these
big
environmental
honeybee
changes affect
enthusiast,
wild pollinator
but when I've been
populations.
going to
What
meetings,
types
more
of
and more I've been
interventions
seeing
might
titles
augment
like this
abundance
at
of
scientific
wild
meetings, like
pollinators.
conserving honeybees
And of course,
does not
interventions
help
would
wildlife.
also
So there's actually
benefit fairly
a lot of
honeybees.
a lot of
and
folks
I'm
a big honeybee enthusiast.
But when I've been going to meetings,
really
more and more I've been
concerned
seeing
about
titles like this.
you probably all talked
meetings,
about this,
like conserving honeybees does not help wildlife.
So there's actually honeybees
a lot of
wild bees.
A lot
so
of folks
that
seems
all the more important that we have enough green spaces and really concerned about
that function as
you
food
probably
for
all
both
talked about
wild
this
and managed
as well,
and also that these
about honeybees
outcompeting
wild bees.
persist.
So of course it seems all the more important that we have
the
enough
way
green
that
spaces
I study and my lab studies
ways
that
to
function as food for
increase
both wild
positive
and
feedback
managed
between human
pollinators
dominated and
also
natural
that
areas,
restored
is
habitats
by
persist
studying
through
how
time.
pollinator foraging behavior.
So the way that I study
the drivers
in my
of
lab
pollinator
studies
foraging
ways
behavior.
to
And whether
increase positive
pollinator
feedback
foraging
between
behavior
human-dominated
changes
and
in
natural
urban
areas,
areas,
and if so, why?
Is by studying how pollinator foraging behavior.
Here we are in the Bay,
studying
and
the drivers of pollinator foraging behavior.
I was trying to find
and whether
I don't have any
pollinator
foraging
behavior
changes in
I
urban areas, and
if
want
so,
to start
why?
work in the East Bay as well. So I'm curious if there are good places that you
here we are
have
in the Bay,
in your
and
I was actually
good.
I was trying to find. So I don't have any sites.
A photo that
was
looking
north from San Francisco,
want to
across
start
the Golden Gate, because I have sites in Marin
and
on the peninsula currently,
but they're all
copyrighted,
have,
of course, because like the
near
Golden Gate is this spectacular view,
So
and
I was trying to find
towards
a photo that was looking north from San Francisco
across the Golden Gate, because I have sites in Marin and on the
my
peninsula.
lab is using these,
but they're all
green
copyrighted,
spaces,
of course, because like the
the
Golden Gate is
a
spectacular view, and so now we're looking towards the beautiful
to study how
urbanization might affect plant pollinator interactions. And I mentioned the angle that we're taking.
My lab is using
by
really
green
trying to
spaces
understand,
like
the
Presidio, Golden Gate Park,
how
pollinator foraging choices
to
study
whether
how
plant
urbanization
species
will
affect
persist
pollinator interactions
in small fragments.
And I mentioned the angle that we're taking.
The project I'll tell you
really
about
trying to
today was
understand
motivated by two things mainly.
how pollinator
foraging choices
three things
affect
the
actual
whether
data that
plant
I will show you is motivated first by the students in the class, really wanting to continue
the
work,
project I'll tell
work
you
in my
about
lab,
today
which
was
is
motivated by
special and
two
delightful
things mainly.
But
I guess
the second
three
reason
things,
was
like
the actual data that I will show you is
repeated
motivated first by the
observation
students in the class, really wanting to
declines
continue
work
plants and
work
pollinator
populations.
So in
special
many
and
cases,
delightful.
we see these declines that
But
are correlated.
the
We don't
second
know
reason was
what's driving what. So is
this
it
repeated
declines in
observation
plant populations
correlated
to pollinators or
declines
vice
in
versa,
plants and
but so really it's been a really
populations.
striking pattern that's
So
been
in many
observed.
cases we see these declines
And then
that are correlated.
the
We don't
second
know
has to do
what's
with
driving
restoration
so
efforts.
declines in plant populations.
So if we think about the continuum from
degraded habitat to restored habitat,
really
of course,
degraded habitat
would be things like
a city with no
and then
real
the second
green
reason
spaces, or a park
has
that
to do
doesn't
with
really have any forage for
restoration
pollinators, or has like a bunch of wood
So if
chips,
we think
but
about the
really nothing
continuum
for the
from
pollinators to
degraded
eat, or,
habitat to restored habitat,
and of course degraded
nesting
habitat would be
areas.
things like a city with
if we think
no
about
real
transitioning
green
those
spaces,
to,
or a park that doesn't really
nice
have any
restored
forage for
areas,
pollinators, or has a bunch of wood chips.
that process is
really
really
nothing for the
important. But
pollinators
I
to
was
eat, or
and I don't think
areas.
appreciate
how tenuous
think
it
about
can be
transitioning those.
So when I first came to USF,
nice restored areas,
I was chatting with Jason Listenby at the Presidio Trust.
That process is
and
really
he's been leading all these
restoration
efforts around San Francisco.
I don't think
and he mentioned that sometimes
how tenuous
plants take hold and
sometimes
the plants
So
don't.
when I first came to USF,
and I thought that was really interesting.
I was chatting with Jason
and I started thinking about
what actually is the mechanism.
And he's been leading all
why
these
there might be
restoration
efforts
around
San
among
Francisco.
restoration projects.
And he mentioned that
why
sometimes
might
the
there
plants
be
take hold and
differences
sometimes
in
which
which
plant
don't.
species actually take
And I
hold.
thought that was
persist
really
over
interesting.
generations.
And I started thinking about, well, what actually is
of course,
the
this has
mechanistic
really
basis
important
for why there might
implications
be
for
variation
the persistence
in
of
native
success
pollinator
among restoration
projects
projects,
and food
or
resources for them.
why might
wild
there
and
be
managed
differences
in
in
which plant species actually take hold and persist over
so,
generations.
what might
and which
lead to
don't.
variable success of urban
Of course,
restoration
this has really
projects,
important
and of course, there are
implications for
myriad things.
the persistence of native
but
pollinator
one that I'm really
communities
interested
and
in
food
exploring
resources
is
for
the role that
wild
pollinator
and
foraging
managed
choices
might
do.
actually have in facilitating plant persistence.
So, what might lead to
let's talk a
variable
little bit
success of
about
urban
how
restoration
pollinator
projects,
foraging
and of course, there are myriad
might
things.
affect
But
fitness.
one that I'm really interested
that
in
is exploring
the
is
number
the role
of
that
seeds
pollinator
produced by
foraging
plant
choices
that can
might
actually
actually have
propagate the
in
next generation.
facilitating plant persistence.
So let's talk a little bit about as bee enthusiasts, I'm guessing that you, like me, usually think about things from the pollinator perspective. The number of
but let's think about
things
things
from
from
the
the
plant
plant
perspective,
perspective,
just
just
briefly.
the next generation.
I'm going to show you a figure from Barry Brosey and Heather Briggs,
So as bee enthusiasts,
I'm guessing that,
and
you, like me, usually think about
pollinator
things from the
foraging
pollinator perspective. But let's think about things from the plant perspective.
Barry
was a professor. I'm going to show you a figure from Barry Brosey and Heather Briggs, 2013 PNAS paper.
The premise
and
is that
really shows how
outcrossing
pollinator
plants
foraging behavior
need to
affect
receive
plant
pollen
seed
from individuals
cells.
of their same species.
So Barry
to
was a professor
set
at Emory
seed,
when he did this
and for
study.
population
Now
to
he's
persist
at
over
University of
generations.
Washington. So here we have a bumblebee,
the premise
which
is that
visiting
outcrossing
delphinium,
plants
and those arrows
need to
indicate
receive
the
pollen
foraging
from
individuals
choices
of their
of the
same species. So you
species.
can see this
In order
bumblebee, she
to
visited
set
3
seed,
delphinium, and then she's carrying
and
a
for
bunch of
population to
delphinium
persist
pollen
over generations.
So
And when
here
she
we have a
deposits that
bumblebee,
delphinium
which
pollen
is
on the stigma of the
visiting
plant, that
delphinium,
plant
and
sets
those
seed.
arrows indicate the foraging choices.
However,
of this
let's
bee.
consider
So
this
you can see
other
this
bumblebee.
She
So in this
visited
case,
3
this
delphinium, and then
is
she's carrying a
switching
bunch of
plant
delphinium
species,
pollen.
and she's
not showing.
and when she
floral
deposits that
constancy,
delphinium
so
pollen
she has
on
a
the
bunch of
stigma of
different
the
types
plant,
of
that
pollen
plant
on
sets
her
seed.
back. And what Brosey and
However,
Briggs
let's consider
showed
other
was
bumblebee.
that
So in
this
this case, this bumblebee is
transfer
switching
of pollen
plant
of different
species, and
species
she's
can reduce
seed
not
cells.
showing,
And that's
floral
been documented by
constancy.
a lot
So
of
she has a
studies.
bunch of different types of pollen on her back.
And so,
And
I'm
what
going to be
Brosey and
using
Briggs
some
showed
terms for the rest of
was
this talk.
that
I'm going to
this
use
conspecific
transfer
pollen
of
and
pollen of
heterospecific
different species
pollen,
can reduce
so
seed
conspecific
set.
pollen transfer,
And that's been
meaning
documented by a
transfer
lot
of
of
pollen of the
studies.
same species, and heterospecific meaning the transfer of
And
pollen
so
of different
I'm
species.
going to be using
onto the
some
stigma
terms
of
for the rest of
plant.
this talk. I'm
So
going to
there's
use
conspecific pollen and heterospecific pollen.
and so I've been really
pollen transfer,
interested in
meaning
whether
transfer of pollen of the same species,
conspecific
and heterospecific meaning the transfer of
pollen
pollen
amount
of
different
bees
species
differs
onto the
among
stigma
sites in
of
the
the
Bay Area,
plant.
and if so,
there's
why
heterospecific pollen transfer.
And so
I've been really
why might
interested in whether
and another way
of saying conspecific
pollen
transfer
amount on bees differs
foraging
among sites in
preferential
the
area,
and
plant
if so,
species is
why?
saying pollinator loyalty.
So by pollinator loyalty, I really mean specialization of a
why might
pollinator
it?
on a particular plant species.
And another way of
why
saying
might pollinator
conspecific
loyalty
pollen
differ
differ
among
among
urban and
conspecific
natural sites? Foraging preferentially on one
let's just
plant
shift our
species
focus
is
back to
saying
that
that
of
of
pollinator.
pollinator.
And
so by
think about
pollinator
what life
loyalty,
as an
I
urban
really mean
pollinator is
specialization
like.
of a pollinator on a particular plant.
This figure is from
So
a review
why might
paper
pollinator loyalty
by Tina
differ
Harrison
among
and
urban
Rachel
and natural
Winfrey,
sites?
who's a professor at Rutgers.
Let's just shift our focus
and
back to
it
that
highlights
of a
how
pollinator
patch
and
sizes
think
of
about
bees
what
of
life
different
as a
body
urban
sizes
pollinator is like.
It highlights how the patch sizes
this figure
that bees
is
of different
from
body
a
sizes
review paper
might be able
by
to
Tina Harrison and Rachel
might differ
Winfrey, who's a
depending
professor at
on where
Rutgers.
nests are.
So that's
and
the letters
it highlights
and
how
big the
patch
patches
sizes
are.
of
So
bees
those are
of
the
different
grade
body sizes
areas.
and so
It highlights how patch
in this
sizes
case that bees of different body sizes
if you have
might
a
be able
patch
to
size that's really big,
might
so you
differ
have like a bunch of
depending
habitat,
on where
that's
nests
gray,
are. So that's
and the
letters
letters
in the
and
middle.
how big the
Both
patches
large
are.
and small
So those
bees
are
can
the
be
grade
supported because there's enough
areas.
habitat for the
And
large
so
bee to
in this case,
get enough energy. And
if you have
there's
a patch
enough
size
habitat
that's really big,
within
so you
the
have like
foraging
a
range
bunch
of the
of
small
habitat,
bee,
that's this gray,
for
and the
small
nest
bee
is in
to
the
be able to
middle,
persist.
both large and
And
small
then here
bees
the
can be
small bee
supported, 'cause
can
there's enough
habitat
use this
for the large
small
bee
amount
to
of habitat,
get enough energy.
a large
and
bee can't persist because
there's enough
there isn't
habitat
just- there aren't
within the
enough
foraging
flowers,
range of the small
to,
bee
for the small bee to be
allow
able to
that
persist.
larger bee
And then
to
here the small bee can
have
use
brood.
this
And then here
amount of
you have
habitat,
but large bees
a large
persisting
bee can't
because they
persist
can
because,
fly far enough.
there isn't
to
just
get
there aren't enough
to
flowers,
these little chunks of
to
habitat, but
small bees can't because they have
allow
smaller
that
foraging
larger
ranges.
bee to then larger bees have brood.
And so
and then here you have
this figure. The purpose
large
of
bees
this
persisting
figure,
because they can fly far enough
to
get
in
to
their
these little
paper
chunks of
was
habitat
to
but
show how
small
patch
bees
size
can't
might relate
because
to
they have
bee
smaller
size
foraging
and the
ranges
bees that could
than
actually
larger bees
persist in particular
and
types of
so,
habitat.
but I think
this
it's
figure, the
really
purpose of this figure,
interesting
for another reason, which is that it really
in their
highlights
paper
how
was
important
to show how
energetic
patch size
costs
might
of travel
relate to
are
bee
for
size
and the bees that
and
could actually
this is
persist in
this is like a
particular types of
conceptual
habitat.
figure, but
is there
I
actually
think it's
evidence
really
that
interesting
for
that
another reason, which is
energetic
that it
costs
really
of travel
highlights
are important and
how
that they
important
might differ
energetic
between
costs
urban
of
and
travel
natural
are
areas.
for pollinators.
And
This
is
is like a conceptual
figure
but
is from
there
Andrew
actually
and
evidence
colleagues
that
this figure we can actually
the
see
energetic
that
costs
the
of
time
travel
that
are
bees
important
spend collecting
and
food
that they
from
might differ
flowers
between
from
urban and
individual
natural
flowers
areas
is longer within small patches
So
of habitat
this,
in urban
areas. This figure is from Andrew and colleagues. So the duration this figure, we can actually see that the time that bees spend collecting food from flowers, from individual flowers, is longer within small
and their
patches of
explanation
habitat
was
in urban areas
that
than in
this
natural
pattern
areas.
of longer
So
foraging
the duration
time
for
for
individual
flower,
flower is much.
the reason for that is that
Longer
bees
for
longer
pollinators
at each
foraging in
individual
urban
flower to fully
areas
extract
than
all the nectar
pollinators
from that
foraging in natural
and recoup
areas.
the energetic cost.
And their explanation
traveling
was
to that
that
patch.
this pattern of longer foraging,
And this leads to the
time
prediction
for
that
flower,
urban pollinators might also fly
the reason
less
for
far
that is that
between
bees
flowers
must
when
longer
foraging.
at
And I
each
don't know if
individual
that's
flower to
true, but I'm
fully
really
extract all
interested
the
in
nectar
it.
from that
And I'm curious if
and
many of
recoup
you
the
know that.
energetic costs.
And it also
traveling
leads to
to
the
that
hypothesis
patch.
that if plants of different species are intermixed,
And this
then
leads to the
pollinators
prediction
might
that
be more
urban
likely to
pollinators
switch
might
plant
also
species
fly
so they
less
can
far
forage on
between
close
flowers
when foraging. And I
but
don't
on the
know
other
if that's
hand, it
true,
leads
but
to
I'm
the
really
alternative
interested in it.
hypothesis that
And I'm curious
even
if
if
you know
pollinators
that.
come across unrewarding flowers,
And it also
they may
leads to the
be
hypothesis that if
likely to
plants of
forage on
different
the same
species are
species
intermixed,
because of the
then
energetic
pollinators
cost of
might be
learning how
more
to
likely to
manipulate
switch those
plant
new
species
flowers
so they can forage on close
flowers.
So,
But on
in
the other
thinking
hand, it leads
about
to the
the
alternative
importance
hypothesis
of
that even if
foraging,
pollinators come across unrewarding
and
flowers,
energy
they may be
costs,
likely to forage on the same
along
species
with my
because
students,
of the
was first
energetic
just
cost of
interested
learning how to
in
manipulate those
documenting
new
the patterns
flowers.
of
Whether pollinators
So,
are
in thinking
more
about the
specialized in
importance
particular
of
sites, and if so, why?
foraging,
And does
and
urbanization itself have
energy
anything
costs,
to do with this?
I,
Along with my students,
So,
was first just
a
interested
lot of
in
researchers
documenting
will
the
follow bees
pattern
around,
of
which is really fun,
whether
but it's also
pollinators
highly error
are
prone,
more
and
specialized
so we
in particular
decided
sites, and if so,
to
why?
actually
And
characterize
does
the
urbanization
pollen
itself
on the
have
backs
anything
of
to do with
bees.
this? Instead of following bees around to
So,
figure out which
a
flowers
lot
they
of
were visiting.
researchers will follow bees around, which is
So
really fun,
now I'll
but it's
introduce
also
you to
highly
the specific
error-prone.
questions
And
that
so we
we have
decided
been asked
to actually characterize the pollen on the
So
backs
with
of
this
bees
instead of
in
following
mind
bees
that
around to figure out which
pollen
flowers
transfer
they were
is
visiting.
much better for plant seeds than heterospecific.
So now I'll
pollen
introduce you
transfer.
to the specific
The first
questions
question that we've been
we
asking
have
is,
been asking.
does the proportion of conspecific pollen
So
differ
with this
among sites in the Bay Area?
in mind that
Conspecific
and
pollen
the second
transfer
question
is much better for
is
plant seeds
what
than
ecological factors
heterospecific
influence
pollen
that
transfer.
proportion.
The first question
And the
that
things
we've been
focusing on,
so it would be
does
really
the
great
proportion
to
of
focus
conspecific
on to do
pollen
kind of like a landscape
differ among
level
sites?
analysis.
We haven't done that, so we haven't been
And
including
the second
like distance
question
between urban
is
patches.
what
But we
ecological
have been
factors
focusing on
influence
things
that
like number of
proportion.
plant species
And the things that we've been
presence of
focusing on,
invasives,
so it would be
total number
really
of
great
flowers,
to focus
to
and
do kind of like a
pollinator
landscape level
identity.
analysis. We haven't done that, so
particularly
we
whether
haven't been
pollinators are
including
common or
like
rare.
distance between urban patches.
So it's
But we
time for
have
me
been
to
focusing on
introduce you
things
to
like
my
number of
sampling
plant
locations
species that you know a lot
presence of
about
invasives
already, I'm
total
sure.
number of
One of them
flowers,
is Azalea Hill, which
and
is so beautiful
pollinator
right now.
identity.
So here
Particularly whether
here's the
pollinators
bay
are
again,
common or rare.
and we have natural sites.
So it's time for me
represented
to
by circles
introduce you
and
to my
urban
sampling
sites represented by
locations
squares. That you know a lot about already, I'm sure.
And you can
One of
see
them
right
is
away that a
Azalea Hill,
limitation
which
of
is
this
so
study is
beautiful
that the
right now.
urban sites are clumped,
So
and they
here's
also tend to be in
here's
wetter
the bay
areas.
again,
So that could be a
and
total
we have
confounding
natural sites
factor
in this, represented by circles, and
a
urban
paired
sites
approach would be
represented by
much better.
squares. So we're thinking about how to what
And
kind
you
of
can see
design
right
to
away
use
that a
in the
limitation
future.
of this study is that the urban sites are clumped.
But
And
for now,
these were the sites that we went to.
so that could be a
did
total
the
confounding factor in
work
this, and
that
generated the data
paired
that I
approach
have
would be much better.
analyzed
So
in the
we're
last
thinking about how to
few weeks, what
for
kind of
you
design
all,
to use in the future.
in 2019.
But
For now, these
no
were
one
the
did
sites that we
last
went to,
summer.
and we did
So
the
here's
work
that
here
generated
are
the data
some
that
examples
I have
of
analyzed
the
in the
site
last,
types. So this is the natural
few
site
weeks
up top
for
and
you
this is
all
Mount Sutro, so
in
an urban site
2019
on the
bottom. And this is part
So,
of the
'cause
field
no one did
team, and this
anything
is
last
Shiso,
summer.
my dog, who you
So
may have heard barking
here's
recently.
here
So
are
the first thing
some
we
examples
did
of the site types. So
was
this is the
we
natural site
characterized
up top,
the
and this
plant
is Mount
community
Sutro, so
at
an urban
each
site
site.
on the bottom.
And this is part of
maybe
the
before
field
I
team,
say
and
that,
this
I
is
should tell
Shiso,
you
my
that the
dog, who
urban
you
sites,
may have heard barking recently.
the diameter of an urban site spans
So the
less
first
than
thing we
4 kilometers,
did
and the diameter of a natural
was
site,
we
like,
characterized the
continuous
plant
habitat
community
spans
at each
more than
site.
60 kilometers.
And
so there really was
maybe
a big
before I
difference
say
in
that, I
patch
should
size.
tell you that the urban
even
sites,
though we were not
explicitly
the
included
diameter of an urban site spans
so the
less
first
than
thing we
4
did was
kilometers, and the
we
diameter of a
characterized
natural site
the
of
plant
continuous habitat spans
And we did
more
that
than
by
60
surveying
kilometers.
plants along
So there really was a
90-meter
big difference in
transects.
patch
So we
size.
just counted all the flowers
even though
and
we were not
identified them to
explicitly
species
including
that were within 15 meters of a transect.
So the first thing we did
15
was
centimeters,
we characterized
15
the
centimeters of the
community.
transect tape.
And we did that by surveying plants along 90-meter
And so in this case,
so we just
this
counted all the flowers and
picture on the
identified them
left,
to
like this
species
is
that
many
were
more
within
15
15
meters
units
of a transect
then 15 centimeters, that would be a lot of work. 15 centimeters of the transect tape,
amount of energy that,
and we counted floral units. And so in this case, this and then picture on the left, a bunch of pollinators, many more floral units. So than a flower like this. How do we actually do the collection? Really, the goal is to catch 50 pollinators per site. Pollinators can get. And we caught pollinators only if they were touching the reproductive parts of the plant. And then we caught a bunch of pollinators. So they were actively foraging on nectar or pollen. How did we actually do the collections?
We brought them
the
back
goal
to
was
the
to catch
lab
50
to analyze
pollinators per
pollen
site.
loads.
And so let's say, for example, we caught a honeybee
if they were
and
touching the
Yampa,
reproductive
and
parts of the
then we
plant.
would capture that pollinator, give it a specimen number, and then we would take fusion jelly and then we
brought them back to
the
the
lab
to
analyze
their pollen loads.
And then we also say, for example, we caught a honeybee and Yampa
fusion jelly to rub off the pollen on
capture that pollinator, give it a specimen number, and then we would take fusion jelly and identify the pollen to plant species that was on the pollinators. And then we also took anthers from the plants.
Here we have a whole bunch
of
slides.
Fusion jelly to rub off the pollen on the pollen from the back of pollinators, so match the pollen of the anthers. And let's say, for example, we saw a situation like this where we have a bunch of pollen from the plant, and so the pollinator was
here we have
on,
a whole bunch of slides.
Some pollen, the back, pollen from the back of pollinators, some from the
made
from
slides
the
from the
anthers
of,
and let's say,
all
for
the
example, we
flowering
saw
plants
a
that we
situation
came
like this
across
where we
have
could
a bunch
actually
of
assign that
pollen
from the
back,
plant that the pollinator was caught on, and we also see some
in this case,
pollen. We would know that this honeybee was foraging both on Yampa and Arigaron.
Made slides
from the anthers of all the flowering plants that we came across. We could actually assign that pollen back. We
to another
captured
species. And
so hymenopterans and
we would know that this
dipterans,
honeybee
but
was
not
foraging
lepidopterans
both on Yampa and Arigaron in the same.
I didn't try to get permits to foraging capture butterflies because there's so many rare butterfly species. So we just have data from, captured hymenopterans and dipterans, but not lepidopterans. And our sample size was 542 pollinators caught on 47 capture butterflies because there's so many rare butterfly species. So we just have data from the students analyzing the slides, and they counted over 200,000
sample size was 542
pollinators caught on 47 plant species. They did so much work. I'm just so
here are
the 158 of the pollinators had fewer than 500 grains, but they counted over 200,000. Decided to just and count 500 grains per slide.
Did so much
and figure out
just so impressed
of the 500 grains how many were 158 of the pollinators had fewer than 500 grains. From a different 335 more than 500
what did we
grains.
actually
so we
find?
decided to just show you is count 500 grains per slide of conspecific pollen differed
of the 500
grains,
sites.
How many were conspecific and how many
here
were,
we have site type
on
from a different species.
X-axis. And
so
then
what did we
actually
find?
The first thing I'll show you is
sites.
And the sites
how
the amount of
green
conspecific
or
pollen
urban
differed
gray.
among sites.
So here we have site type
on the X-axis.
So
and then
here you
can
can
see right away that there was a lot more
and the sites
conspecific
natural and green
pollinators caught
in
urban sites.
And just to remind you,
this was a very statistically robust
So
relationship.
here you can see right away
it's
that
kind of interesting because
a lot
basically what it
conspecific
means, and maybe this is
on
unsurprising to those
caught
of you that
in
have
urban
lived here for a long time.
This was a
very
and,
statistically
watched
robust
pollinators
relationship
in urban areas.
And
But I was actually a little
kind of
surprised
interesting
about this
because
when I was
basically
living
what
in
it
Tucson,
means,
I
and
did
maybe
some
this is
similar work and actually
unsurprising
found
to
the
those of you
opposite
that have
pattern.
lived here.
Essentially,
for
this
a
means
long time. Pollinators in these urban sites
pollinators
are foraging in urban areas. But I was actually a little
plant
surprised
species
about this because
in
when I
natural
was
sites
living in Tucson, I did some similar work and actually found the opposite,
which was
pattern.
really
So,
surprising to me because
essentially
this
this
means
means
that
that
in the natural
pollinators
sites, so
in these urban sites are
so many
foraging
different types of
preferentially
pollen.
on single plant species, whereas in
So
the natural
another
sites,
way of looking
they were
at
carrying
this
a lot of
in
addition to just what proportion of the
which
pollen
was
grains
really
were of the
surprising
same
to me
species on which
because
the
floral
pollinator
abundance
was
was
caught,
pretty high
is
in the
to
natural
figure out.
sites. So I don't
exactly
know
how
why
many
there were
species
so
of
many different types of
plants from how many species of plants pollen.
So another way
that
of
was on
looking
the
at this
pollinators. In addition to just what proportion of the
So
pollen grains were of
I'm
the same
also
species
gonna
on
show
which the
you
pollinator
now
was caught,
naturally
is to
urban
figure out
sites,
exactly how many
and
pollen
of
species richness. So
plants,
how many different
from how many
types
species
of plants
species,
pollen
came
were represented
that was on
in the
pollinators.
pollen loads on the pollinators. So I'm also going to show you now natural and urban sites. So here,
and pollen
these are
species
means and
richness,
standard
so how
errors,
many different
types
first
of plant species
over all the pollinators
within a site, and then averaged over
loads
the
on
sites.
pollinators.
And there were more different types of pollen found on natural pollinators. So here, these are means and standard errors
in urban
sites
over
which
all
again
the
was really
pollinators
within a site and then averaged over the sites. And so we were thinking, well, what could be driving different types? Perhaps there are just way more plant species in natural sites that look similar than. So we assessed whether the number of plant species again was really surprising in the natural sites that we had sampled from. And so we were thinking, well, what could be driving this So now I'm going to show you these are plant data, natural sites that look similar. And so we the number of different plant
whether the number of plant species was higher
in the natural sites that we had
natural sites to urban sites.
So now I'm going to show you, these are plant data that I'm going to show you.
There was
the number of different
statistical
difference
between natural sites,
and we're going to compare
the number of plant species
natural sites to
those
urban
sites
assessed via transect data. But we were thinking, just
there
because a site
was
is urban,
no
characterized as urban,
statistical difference
between natural sites and urban sites.
That might be interesting, but
that were in
those sites,
perhaps plant species richness does actually
data.
drive the amount of
But we were thinking,
pollen, regardless of
just
whether
because
a site is urban or not.
So
characterized
we
as
also
urban. Looked at whether there was a relationship between the number of plant species and the amount of conspecific pollen. Perhaps plant species richness does actually drive the amount of conspecific pollen.
And we found a negative relationship,
regardless of whether a site is statistical relationship. So
we also
higher plant species richness whether there was a relationship between the number of plant species and the lower amounts of conspecific pollen. So if there's more plant species, then you
we found a negative relationship.
More likely
to have
there is a statistical relationship. So
pollen represented
higher
in
plant
body,
species richness is
associated
but it was a
with
pretty
lower
weak
amounts
of
of
conspecific pollen. So if there's
one
more
thing I'll
plant
point out,
species,
I think
then
it's interesting is that there are some sites that
more
have really
likely to
low
have more plant species richness,
pollen represented
but
on
that
your
still
body
have
if you're a
pollinators that
pollinator.
carry really low proportions.
But it was a pretty weak
pollen.
relationship.
So that means pollen from many different.
One thing
species.
I'll point out, I think it's interesting is that there are some sites that have
the
really
fact that
low
pollinators themselves are actually better assessment of the plant species,
but that still have pollinators that carry
really low proportions of conspecific pollen.
or it could indicate that the
pollen
pollinators
from
are
many
flying
different
from other places.
And
this
where
could
they were foraging on different
reflect the fact that
pollinators themselves are actually better assessment of the plant species.
This could mean that
community than a
this could also mean
transect
that
is
low
that human species
do.
richness of
Or it could indicate that the pollinators are
floral
flying in
abundance.
from other places.
And so we also examined
where they
if
were
there's
foraging
a
on different
relationship
types of
between the
plant
total number of
species.
flowers and how much conspecific pollen.
This could
there was.
mean that
This could also mean that
assuming that higher
species
floral
richness
abundance would mean
the transects
would
reflects low
be
floral
associated with
abundance,
more conspecific pollen.
And so we also examined if there's a relationship.
Here I'm going to
between
show
the
you
total
the
number of
relationship
flowers
between
and
floral
how
abundance
much
and
how much
pollen there was.
conspecific pollen there is.
Assuming that higher
I
floral
actually ran the
abundance would
statistics
mean- or
before I
would
graphed
be
this,
associated with more
and
conspecific
I
pollen.
found a really strong
So
positive
here I'm
relationship.
gonna show
And I was
you
like,
the
oh
relationship
wow,
between
that
floral
totally makes
abundance
sense. If there are
and
more
how much
flowers in an
conspecific
area,
pollen there is.
and pollinators are more
And
likely to
I
be
actually
loyal
ran the
to
statistics
plant
before
species,
I
probably
graphed
because there's
this,
more flowers of
and
that plant
I
species.
found
a really
But then
strong
when you actually
positive
look at the
relationship. And I was
data,
like, oh wow, that totally
they look
makes
like they
sense
have
if there
an
are more
outlier.
flowers in an area.
So I think there's
and
actually no
pollinators
real
are more
statistical
likely to
relationship
be
here.
loyal to the
This
plant
outlier
species,
was probably because
Yampa,
there's more
and
flowers
there
of
were
that
just
plant
so many
species.
flowers because we were
But
counting
then when you
individual
actually look
floral
at the
units,
data,
not the stems.
they look like they
Um,
have an outlier.
So I think there's really.
So I think there's actually no real statistical
not a relationship. When we
here.
remove the outlier,
This
the
outlier
relationship
was
disappears.
Yampa,
And there
so
were
the
just
total
so
number
many
of flowers
flowers, so
doesn't
we were
predict
counting the
pollinator
individual
loyalty,
floral
but what
units,
about
not
the abundance
stems.
of individual species?
So I think there's really
of plants. That is,
not a
do
relationship.
pollinators
When we
forage
remove the outlier, the
preferentially on
relationship
species with
disappears.
high relative abundance? So the total number of flowers doesn't
So we
predict
also looked
pollinator
at
loyalty,
whether
but
plant
what about
relative
the
abundance
abundance
predicts,
of
the preference that a pollinator
species,
has for a
of
particular
plants?
plant species.
That is, do pollinators forage preferentially
we,
on species with high relative
generated a
abundance?
metric that reflects
So
pollinator
we
preference.
also looked
It
at
ranges
whether
from 0
plant
to
relative
1,
abundance
and a
predicts,
value of 0 indicates
the
the preference
preference
that a
carried
pollinator
less pollen
has
than would
for
be
a particular
predicted based on plant
species.
relative abundance. A value of
And
0.5
we,
indicates that
generated a
pollen
metric
amounts
that
match
reflects
expectations based on
pollinator
plant relative
preference.
abundance.
It ranges from 0 to
And
1.
a value of
And
1
a value of 0 indicates
means
that the
pollinators
pollinators
carried
way more
less
pollen than would be expected.
predicted based on plant relative abundance. A value of 0.5 indicates
And
that
we use the transect
observed
data
pollen amounts
to
match expectations based on plant relative abundance.
So we just
And
wanted to
a
know,
value of
does
1
plant relative abundance actually
means
predict
that the pollinators
pollinators
carried way more pollen than
for
would be expected
plant species
based on plant relative abundance.
And we found a
And
negative
use the
relationship.
transect data,
So this was really
to
surprising to me. And I'm
generate
very
this
curious
figure.
if any of you have ideas.
So we just wanted to know,
for
does
why
plant
this might
relative
be the
abundance
case.
actually
So
predict pollinator
the more
preference for
abundant a particular
plant
plant
species
species is, the less likely
And we found
the
a negative
pollinators were
relationship.
to have
So
a lot of
this
pollen
was
from that
really
plant
surprising
species
to me,
on
and
it.
I'm very curious if any of you have
It
ideas.
could reflect that
for why
we
this might be
were
the case.
sampling,
So,
the more
after the
abundant a
pollinators
particular
first came
plant
out in the morning,
species
and
is,
perhaps those
the
flowers
less
already,
likely the
flowers of those
pollinators
species
were
were
to have a
depleted of
lot of
nectar,
pollen from that.
so
plant species
pollinators switched
on it.
to other rarer species.
It could
I don't
reflect
know,
that
but, I'm really
we were
curious
sampling,
if folks have
other ideas
after
for
the
why this could be
pollinators
the
first
case.
came out in the morning, and perhaps those flowers were already
the flowers of those species were depleted of nectar, and so pollinators switched
so
to
I've been telling you
rarer
a whole bunch of
species.
statistical relationships
I'm really curious
between, you know, ecological metrics
ideas
and
for why this could be the case.
conspecific pollen. But what about the specific plants and pollinators themselves?
So I've been telling you a whole bunch of
statistical relationships
by the
between,
species,
you know, ecological metrics.
You've just started to kind of slice the data and understand that.
And
you have this massive
data
set
with
all these
interactions,
plants, and
so we can really look in
themselves.
detail. But
At
the moment,
patterns
we're
actually driven
kind of like a high level analysis
by the particular species
so
that are out there.
The two
You've
things
just
that we've
started to
done
kind of
to
slice
really
the data
try
and
to
understand
get at
that.
whether the
identity of
so you have this massive data set
with
all these interactions.
on the
so we
plant
can really
side
look in
to
detail,
look at
but
whether
at the moment
plants are
we're
invasive
this is
or
kind
not.
of like a high level
So
analysis.
I'll tell you that first. And then the second is to understand if
the two things that we've done to really try to get
so in terms of
invasives,
identity of plant or pollinator
the
species matters.
three most invasive
on the plant side to look at whether
we
plants
have
are invasive
in our
or
data
not.
set are wild radish.
So I'll tell you that
mustard,
first.
and
And the second is to understand
all
if
of
the
which are really
pollinators are
important
rare or common
plants for pollinators in this
So in terms
area.
of invasives,
And so
I'm going to show
three
you
most invasive, or the most common invasives
the proportion of conspecific
in our data set
are
are wild
invasive
radish,
and non-invasive
and
and
hairy
urban
vetch,
natural sites.
all of which are really important.
So here we have
plants
natural
for
sites,
pollinators in
urban
this area.
sites, and then I'm
and
going to
so
show you
I'm going to show
the data in
you
bar graph form.
The proportion of conspecific pollen
So
for invasive
what
and non-invasive
jumps
in urban
out
and
right
natural
away,
sites.
I think,
So
is
here we have
once again
natural
we see
sites,
that there's more
urban
conspecific
sites,
pollen in
and
urban sites.
then I'm going to
already
show
knew that.
you,
But in
the
both
data
cases,
in bar graph form.
the pollinators carry more pollen
So
from the species on
what
which they were caught.
jumps out
right
that
away,
species
I
was an
think,
invasive
is
plant.
once again we see that
So
there is more
pollinator
pollinator
preference
in
for
urban sites.
invasive
So we already knew
plants.
that.
But in both
the
cases,
urban and the natural sites,
the pollinators carry more pollen from the species on which they were caught, if that species was
and then
an
on the
invasive
pollinator side, the
So there
slice
is
we've
pollinator
taken so
preference
far
for
is to
invasive
just look
plants,
at rare versus common
in both the
pollinators.
urban and the natural sites.
With the assumption that these patterns could be driven just by what some common pollinators like Apis mellifera.
And then on the pollinator side,
we were
the
interested
slice
in
we've
knowing
taken
whether,
so far is to
if
just
it's
look
really
at
just
rare
like
versus common
honeybees that are driving
pollinators.
this entire pattern.
With the assumption that
honeybees in our data
these
set,
patterns could be driven just by what some common
So,
pollinators like Apis mellifera
here we have natural and
is
urban
doing.
sites,
We were
and
interested in
prevalence
knowing
means whether
the pollinator was rare
if it's
or
really
common.
just like honeybees that are
So the
driving
first thing
this
we had to do
entire
was
pattern,
determine,
'cause we did have a lot of honeybees
which
in our
insects were
data set, of
common,
course.
To all or the majority of sites.
So
And it turned out that there was
here
a really
we have
big difference
natural and
among
urban
species.
sites, and
Most
prevalence
insect
means
species
whether
were found
the
at
pollinator
5
was rare
or fewer
common.
sites.
With the exception
So the
of
first thing
one
we had to do
honeybee
was
and one
determine,
bumblebee species,
which insects were common,
then
to all or the majority of sites,
which were
and it
found
turned out that there
at
wasn't
10
really a
and
big
11
difference among
sites,
species.
respectively.
Most insect species were found at 5
and so
or
I'm going to
fewer
show
sites.
you,
So
with the
basically
exception of
common
one
now
honeybee
means
and one bumblebee
either
species, so
honeybee
of course,
or
and then
that
Bombus
common
phasonsensia,
bumblebee, Bombus phasonsensia,
which were found at 10 and 11 sites.
So I'm going to be
respectively.
showing you the amount of conspecific grains
and
for
so I'm
rare
going
and
to show
common at
you,
both types of
so
sites.
basically common now means either honeybee,
And we
or
do
that
see that
common bumblebee,
common
Bombus phasonsensia.
species carry more
And
conspecific pollen.
So I'm going to
Kinda
be showing
interesting.
you
So
the
they're
amount of
carrying more
conspecific
pollen
grains for rare and
of
common
the
at
plant
both
species
types of
on which
sites.
they were caught. And that moving
And we
from
do see
natural to urban
that
sites,
common
increase
species
is much
carry more
higher
conspecific
for common
pollen.
pollinators.
Kind of
They're much
interesting.
more
So they're
likely
carrying more
to carry
conspecific
of the
pollen
plant species on which they were
at
caught.
urban sites, and it's not the case
And that
for the
moving
rare
from
pollinators.
natural to urban sites, the increase
I am
much
not quite sure
higher
how to
for
explain
common
this yet.
pollinators.
I thought
They're
a little
much
bit about
more
it, but
likely
I'm also
to carry
curious your
conspecific
thoughts.
pollen
At urban
so
sites.
those are all the data that
for
we have
the
generated
rare
so far.
pollinators.
And so just to remind you, which
I'm
I'm not
does the
quite sure how to
proportion of
explain this
conspecific
yet.
pollen
I
differ
thought a
among
little bit about it,
sites?
but I'm also curious.
And we found
your
that
thoughts.
pollinators carried more conspecific pollen.
Okay, so
in
those
urban
are all the
areas.
data that we have generated so far.
And then
so
we
just
found,
to
we
remind
asked,
you, what ecological
we were
factors
asking, does the
influence
proportion
the
of conspecific
pollen
differ
pollen
among sites?
And we found that
pollinators
pollinators
carried more
conspecific pollen
in
plant
urban
species
areas.
at sites with high plant species.
And then we
asked what ecological
species
factors
richness.
influence the proportion
So
of
if a plant was
conspecific
common,
pollen,
then there was less
and we found that pollinators carried less,
and there was a
conspecific
negative
pollen
relationship
from
between
common plant
the
species,
number of plant
at
species
sites
in a
with
community
high
and
plant
species
pollen
and at
amount.
sites with high plant species.
Both of those I think
richness.
are really
So if a plant was common, then there was less conspecific pollen. And then we also found that pollinators carried more conspecific negative relationship between the number of plant species in a community, and somewhat less surprising. We know in many cases pollinators actually do both of those. I think are really and they carried more conspecific pollen if they were one of. And then we also found that pollinators carried more conspecific pollen. All right. So what does this all mean? What are the take homes? Somewhat less surprising. We know in many cases pollinators actually. I think the first take home is that restoration can be meaningful both in terms of how it makes landscapes look and also in terms of the ecological functioning. That is, what we found in terms of the pollen loads translates into pollen deposition on the stigmas, then that may. I think the first take home is that restoration can be meaningful both in terms of how these results have also inspired us to think about how plant species distributions influence. That is, what we found in terms of the pollen loads translates into pollen deposition on the stigmas. So of course, many natural areas are invaded with invasive grasses, for example, which can result in individuals. And these results have also inspired us to really about how plant species distributions influence the patterns of like individuals of the same species. And in urban fragments, so of course many natural areas are invaded with invasive grasses. So we're investigating the role individuals of plant distribution being really far away from each other in the amount of conspecific pollen, like individuals of the same species. And then we've also started urban fragments that pollinators among urban fragments to really understand the longer so we're investigating the role of urbanization, the plant distribution within. If levels of connectivity among urban fragments are low, even if conspecific, and then we've also started thinking about the movement of pollinators among urban fragments within pollinator and eventually the longer-term consequences of urbanization, determining both the quantity and also levels of connectivity among how genetically diverse the pollen is. Even if conspecific pollen transfer occurs and plants can produce seeds, inbreeding within pollinator and eventually plant populations. Okay, so with that, I'll thank you so much for quantity listening to this talk, like how and I will acknowledge the University of San Francisco Faculty Development Fund, which funded we're looking into research and allowed us to buy things like nets and translate tape. Okay, so with that, I'll thank you so much for that worked listening to this talk. And I will acknowledge the University of San Francisco Faculty Development Fund, which funded this research and allowed us to buy things like nets and
Peter, I have
tape.
A question right off the bat.
I'll acknowledge the wonderful students that worked with me, and
monoculture being bad for the pollinators or for the bees. Like when you go down to Bakersfield during almond season, all you see is Peter. I have a question right off the bat. Flowers. Yeah, you know how we always talk about and. Monoculture being bad. So this kind of indicates pollinators or for the bees, like when you go down to Bakersfield during almond season, all the diversity of almond, white pollen. Even if they're stuck in the city, they're going to look different. And that's interesting. So this kind of indicates I didn't know the bees knew enough are worried about nutrition to diversify their plate of. So they probably do better than we do. Stuck in the city, they're going to look at different.
Yeah, it's really interesting too because that's a really good point. Like
because it's I didn't know the
a diversity of plants. A lot of them might not
to diversify their plate
versus in natural areas.
So they probably do better than we do.
There may be more. Yeah, it's really interesting too because that's a really good point. Like in urban areas, even if there's a diversity of plants, a lot of them might not actually be good resources for the bees versus in natural areas. There may be more plant species that actually are good for bees.
Bees coming to them.
Yeah, that's interesting.
They're getting their pollen out there the most. For me, it seems to highlight how successful invasive.
Yeah, that's a really good point. And I didn't show you the relationship between common pollinators and invasives, like putting it all together, but the common pollinators are more likely to go to invasives too. And so yeah, that's a really good point. And I didn't show you the relationship between common pollinators and invasives, like putting it all together, but the common pollinators are more likely to go to invasives too.
Whatever.
And so yeah, it's positive feedback.
It seems like the timing of when the invasive species are blooming, because I know coincides just looking into invasive species, here in Marin and our native bees might already whatever, lots of things. It seems like the timing of when the invasive species
that's a really good point. And so coincides it will be really interesting to actually do a time series
honeybees, when maybe some of our native bees might already
we just took a snapshot and
because
we started in May, but it's already past the time that that's a really good point. And so it will be really interesting to actually do a time series and look at these relationships over time. We just took a snapshot and we started.
It might not be such a big deal in the area around San Francisco State, but when I think about our friends in Alameda County, star thistle, which is a late bloom. It's more of a summer bloom. And I guess, maybe in San Mateo County, such a big deal in the area around San Francisco Marin, you start, like Nevada, you start getting star thistle. I think about our friends in Alameda County, that's going to be interesting because, our native bees will be kind of summer bloom done, right? And I guess, maybe in San Mateo County, you have some star thistle, and when you start going north, yellow star thistle, Marin, you start, like in Nevada, you start getting star thistle, but that's an invasive. That's going to be interesting because, our native bees will be
and I'm wondering, Sevan, if you have any,
right? And then all of a sudden you have this new big honey flow,
like what Peter was talking about,
thistle just kicking in.
There's a lot of like big commercial beekeepers who really, I think, judging by the timing if you have any bees are here and not here, they're making their money off of almond pollination and maybe a secondary pollination. But they bring like 100 to 200 hives back to a site at a ranch. Really, I think, judging by the summer when we've got nothing here and not here, they're making their money off of almond pollination, sugar syrup, maybe a secondary pollination. But the only thing blooming, then they bring like cows aren't grazing down at back to a site at a ranch in West Marin. So I'm curious what you think about like the pollination, like how we're destroying ecology, sugar syrup on all the hives. But the only thing blooming that the cows aren't grazing down at that time is the star thistle increasing. So I'm curious what you think about like the pollination, invasive species, like how we're destroying.
Yeah, that's so interesting, right? The honeybees are then just facilitating the propagation of the invasives, but they're the only things there that are flowering
increasing. Yeah, the invasive species in other areas.
Yeah, that's so interesting, right?
How you compensate for the
then just facilitating the propagation of the invasives,
I'm wondering how you
but they're the only things there
time of day.
Are flowering at that
various
time.
Species give off nectar at different times of the day and open the male. I'm wondering how you compensate for the and yet when you're out collecting, you compensate for time. You've got a lot of sites to go to. You can't various species test them, give off nectar over a 6-8 hour period of the day and open the male and
that's
female
such a good point. Yeah, that's a really good point. I mean, we did it one site per day, so we're at the site for a long time.
You've got a lot of sites to go to. You can't
a lot of plants will have been depleted of nectar, and even if it's replenished like slowly, that's such a good point. Yeah, that's a really good point. I mean, we did it one site per day, so we're at the site for interactions like the pollination network. But even then, a day, a lot of plants will have been depleted of nectar, and even if it's replenished like slowly, it's an excellent point. But the proportions of pollen and interactions, like the pollination network can change just
were the European honeybees in the list of common pollinators? This really just shows
a snapshot in time. Yeah, so there was the European honeybee, and then it was Bombus cosmosensis, so just like one honeybee and one bumblebee.
I'm just curious. Got it. Were the European honeybees in the list of common pollinators? Well, I guess either in the urban or in the natural.
Yeah, so it was the European honeybee, and then it was Bombus cosmosensis, so
more
just
of one
like
than
one
the
honeybee
other.
and one bumblebee.
I don't know. One of the reasons I'm asking is so literally just today at the camp where I work, I guess either in the urban or in the natural. Smells wonderful. And were you finding that you were capturing it filled with honeybees and it was more of one than the other? And I don't know. One of the reasons I'm asking is I couldn't find literally just today anywhere on there where I work. And I think to David's point, depending on the time of day, different species might open up at different times. And even have more sugar. I couldn't find one honeybee anywhere on there. And I'm just curious if you noticed any kind of correlation between depending on the time of day, different species might open up at different times. And even have more sugar, for instance, early in the morning than later.
So it was basically,
and I'm just curious if you noticed any kind of correlation
between
the
bumblebee was found at 11 of the 12, and the honeybee was found at 10 of the 12, and all the rest were found at fewer than 5. That's an interesting point. I think, and I do think that there was like one site that we had 12 sites had bumblebees and not honeybees, and one that had bumblebee was found at 11 of the 12, and the one that didn't have 10 of the 12, and all the rest were found at fewer than 5. I think was an urban site. And I do think that there was like one site that had bumblebees and not honeybees. That's all I can remember off the top of my head, but I'm totally happy to find out. And the one that didn't have honeybees, I think. Ali, thank you. Should I answer some of the questions in the chat? Interesting. But yeah, that's all I can remember off the top of my head, but I'm totally happy to find out, look at the data. Oh, was there much overlap between plants in urban and rural transects? Ali, thank you. Should I answer some of the questions? They were pretty different, but there was some overlap. Like, there were poppies
in both types of sites. The invasives were similar. I would actually say the invasives were more similar.
Oh, was there much overlap between plants in urban and rural
between
transects,
urban
or
and
the
natural
plant
sites.
population is very different?
They were pretty different, but there was some overlap. Like, you know,
they were restored
poppies
with
in
native
both
plant
types of
communities, so they
sites. The
did have a
invasives were
bunch of
similar.
native
I would
plants,
actually say the
but,
invasives were more
there were,
similar
like,
than the non-invasive
Clarkia
plants
and
between urban
some
natural
species
sites.
of plants that were really,
I was only
the urban
seeing them
sites,
in the natural sites. Although I know they have
were
been,
restored
with
with native plant
the
communities, so they
Presidio
did
Trust. I
have a
haven't
bunch
seen
of
it there
native
yet.
plants, but
So
there were like Clarkia and some species of plants that were really, I was only seeing them.
I haven't done
natural
also. I
sites.
haven't
Although
done.
I
Like,
know
a
they
network
have been
analysis
like
yet
restored
to look at
in the Presidio Trust, but I haven't seen any there yet.
plant pollinator networks.
So, but I should make a Venn diagram of. That's a really good idea of like the plant species that
I haven't done
looking
also.
for.
I haven't done like a network analysis yet to look at. And then look and see if like it's actually the individual.
But I
plant
should
species
make a
potentially
Venn
driving the
diagram
pattern.
that's a really
Like
good
if there's
idea
more
of,
conspecific
like,
pollen on
plant
particular plant
that were
species,
present in both.
and there are just fewer of those in natural sites.
Besides
And then
the
look
invasive.
and see if like it's actually the individual plant species that were
Bonnie, are
potentially
you
driving
muted?
the pattern. Like if there's more conspecific pollen on particular plant species, and there are just fewer of those in natural sites. Besides the invasive. Bonnie, are you muted?
Hi, I was just wondering, in your choice of urban areas, did you pick sites that were purely native plants versus plants that were non-native? I was just wondering, in your choice
sites
of
that
urban areas, did you
contain
pick
restored plant communities?
sites that were
So we didn't go to like urban gardens that had ornamentals. We only went to places that had been restored. You picked sites that contain restored plant
Dan was asking, do
so
you
we
have a
didn't
metric for
we didn't
flower
go to
density,
like
not just plant diversity?
urban gardens that had ornamentals.
Flower
We only
density,
went to
yes.
places
So,
that had been restored.
Well, we have sort of. We have like number of flowers that were present along a 90-meter
Dan was asking, do you have a metric for flower density?
And then Jerry
not
asked,
just one would expect in urban transects there would be
flower
water
density,
areas
yes.
versus
So,
dry transects
well,
in late
we
summer
have sort of
in rural areas.
We have like
So
number of
how do you
flowers that
think
were present along a 90-meter transect.
impact?
I think that is really
Jerry asked,
important, and I
one would
think that's
expect
probably
in
the
urban
biggest
transects
confounding.
there
I
would
think
be
it's a great
watered
point. I think
areas
it's probably the
versus
biggest
bone-dry
confounding
transects
variable
in
in
late
all of
summer
this. So I think
in rural
if
areas. So
if
how
this
do you
is
think
primarily, it's possible that this is primarily moisture driven.
I think that is really important, and I think that's probably the biggest confounding. I think it's a great point. I think it's probably the biggest confounding variable.
It's not urbanization per se. It just happens to be the characteristics of the urban
this is
landscape
primarily,
that we have
it's
here
possible that this is
versus the
primarily
drier,
moisture driven, such that there's just
natural
more
area.
nectar.
So I think that's something we have
areas
to
because they're more moist.
Really
And that
good
it's not urbanization per se. It just happens to be the characteristics of the urban landscape that we have here versus the drier
Any other questions?
natural area.
I have more
I think
questions for you.
that's something we
but
have to
they're totally
tease apart.
random. I'll follow up offline.
It's a really good point.
Yeah. But if no other questions, thank you so much for being with us and sharing. It's super fascinating, very interesting. I have more questions for you, but we're totally random.
Thank you so much. I love hearing all these questions and thoughts because this is really very.
But if no other
preliminary.
questions,
I think
thank you so
actually
much for being
the
with
moisture
us and sharing.
comment is that's
super fascinating,
that's kind of like
very interesting.
Going to be really important
research.
to actually pair.
Thank you so
urban
much.
and natural
I love hearing all these
that
questions
are sites
and
that
thoughts
are
because this
like
is really very
moisture
preliminary.
And I think actually the moisture comment is that's kind of like going to be really
so
important
many
to actually pair urban and natural populations.
And I look forward to
or sites that are
here in the last
long
couple
moisture
years, so I
gradient
look forward to
instead of having
seeing
all
what
the urban sites be
you do in the future
moisture,
and
'cause
what
obviously
more
it's a
research you have to share with
important
us.
confounding variable.
There's so
Bonnie,
many
thank you,
fascinating
everyone.
variables in all this.
Oh, thank you. Thank you
I look
so much.
forward to, I mean, you just hadn't started here in the last couple of years, so I look forward to seeing what you do in the future.
So we'll let
and what
Sevan
more
go,
research you have to
but
share with us. If anyone, I'm going to log off. I will leave everything open.
Oh,
in case
thank
you guys
you.
wanna
Thank you so
answer,
much.
like
Sevan.
talk to each other.